Welcome to the video for chapter 5.2. We're going to be talking about bacterial membranes and transport. So we're going to start to bring stuff that we learned about in chapter 4 and bring it and start to apply it specifically to a prokaryotic bacterial cell. Okay, so our section objectives for 5.2 is you need to be able to describe the functions of a cell membrane. You need to be able to explain how nutrients are transported and how energy is spent to drive transport. And you need to be able to describe how pathogens use ion gradients and ATP to obtain nutrients from the cell. Okay, let's talk about bacterial membranes and how they move things across. Okay, so just a reminder, the cell membrane, the plasma membrane, the cytoplasmic membrane, remember all of these are names for the same thing. The cell membrane is going to define the cell. It's going to be what we call a boundary. Okay, so it's going to delineate between the interior environment and the exterior environment. Okay, so it's going to be what separates the cytoplasm from the extracellular matrix from the outside environment. Okay, so just a reminder, cell membranes are made from a phospholipid bilayer. So we have this, our hydrophobic interactions going on here. So we have the hydrophilic heads, which are going to be pointed to the outside of the outside environment and towards the cytoplasm. And then we're going to have this area here in the middle. Okay. This is going to be all hydrophobic. So it's not going to want to interact with water or anything that's polar or charged. Okay, so whenever something has to come through the membrane, not only does it have to come through the heads, so that polar region, but it also has to cross this non-polar region. Okay, one of the main jobs, besides being a boundary for the cell membrane, is to regulate what comes in and what comes out. Remember, the membrane's semi-permeable, so some things can go through the membrane and some things can't. Okay, we talked about channel proteins and transporters and other ways that things can come across the membrane. Okay, but this is the main focus of a cell membrane. Okay, so one of the things we talked about in chapter 4 was the makeup of the plasma membrane. So we talked about how it has lipids, so that phospholipid bilayer, the cholesterol to help it stay fluid. But we also talked about these proteins. So we have those transmembrane proteins that cross all the way. We have what we call peripheral proteins that just kind of sit on the surface, whether it's the outside surface or the inside surface. Okay, but these membrane proteins are really important. They have a lot of functions for the bacterial cell. So let's talk about some of those functions. Okay, the first thing is these membrane proteins act as support for what we call the motility structures. These are going to be things like the flagella and the pili, the things that are going to help the cell move. Okay, so these membranes are going to act as a place for them to anchor and support so that they can stay attached to the cell. Okay, we have other proteins. We've talked a lot about receptors and how they can that can communicate with other cells. But one of the things these receptors can do is they can detect what's going on in the environment. Okay, so detecting environmental signals is the next big function for them. Okay, so when a bacterial cell is in its environment, bacteria don't really get to choose where they end up. Okay, so when certain things bind to certain receptors, it's going to indicate to the bacterial cell whether that's a good environment or a bad environment. Okay, if it's a good environment, they're going to stick around and they're going to grow and divide and just flourish. Now, if it's a bad environment, a couple things can happen. Okay, if we are one of the endospore creating ones, we're going to signal that pathway and we're going to shove all of our genome into an endospore so it can survive until the conditions change so that that bacterial cell can survive in that environment. Okay, the second thing that they can do is they can actually just start moving. Okay, trying to get away from that bad environment. So they're going to move a little bit, test the environment with those receptor proteins. They're going to move a little bit, test the environment again. And they're going to continually do this until they sense that they're in a good environment again and are away from that bad one. Okay, we've talked about membrane proteins and their function with moving things across the cell. Okay, so one of the things bacterial cells can do is they can pump things out of the cell. Okay, so they can pump out toxins in the case of Vibrio cholerae and the cholera toxin and other virulence factors. One of the things that is very distressful for us is that some of these bacterial cells have actually evolved that they have pumps that are going to actually take our antibiotics that we're trying to use to kill those bacterial cells, and they will pump them back out into the extracellular matrix so that they can't do them harm and not kill the bacteria. Okay, we can bring in nutrients. That's part of transport we've talked about. Okay, we can take out or exclude toxins like antibiotics. Okay, but one of the most important functions is that they're going to establish these concentration gradients. Remember, nothing can move across the membrane, especially in what we call passive transport, without that concentration gradient. These concentration gradients are really important. So one of the jobs that some of these membrane proteins have is to make sure that there's always a concentration gradient, that it doesn't equal out, and that transport of things across the membrane stops. Okay, we have talked mostly to this point about a specific type of transport. It's called passive transport. Now, if you remember from biology, passive transport is the type of transport that moves things across the membrane with no energy being used to move something across the membrane. Okay, so we've talked about diffusion. We talked about diffusion that goes directly across the membrane, like is pictured here. Okay, and then we've talked about what another form of diffusion, when they use the proteins to go across the membrane. This has a special name. It's called facilitated diffusion. When anything is facilitated, it usually has something that helps it. So in this case, facilitated means we're going to use a protein, whether it either be an open channel protein like this one, or a gated protein that is going to require the solute to bind to the protein in order to get that shape change that it's going to allow it to come across the membrane. Okay, passive transport is the type of transport that really relies on that concentration gradient. If there's not a difference in concentration, aka the concentration gradient, it's not going to move things across the membrane. It's going to just sit there happily in equilibrium. Okay, so we have to have an area of high and then we have to have an area of low to move things about. Okay, and then the final type of active transport, we've talked about this a little bit so far. Okay, we talked about endocytosis and mentioned briefly exocytosis. So endocytosis and exocytosis are sometimes referred to as bulk transport. Okay, both of those are a form of active transport or the type of transport that is going to use energy to move something against the concentration gradient. So with passive transport, it's using the concentration gradient. So it's going to go from a high concentration to a low concentration. Okay, so like here we have the high concentration of purple circles and it's low inside. So they're going to move across the membrane into inside the cell. Now with active transport, because we're using energy, we can move things against the gradient. So we can go from an area of low concentration to an area of high. So if we see here in this picture here, we have the yellow circles and the orange diamonds. Those aren't triangles, orange diamonds. Okay, so what's happening here is this transport protein is moving the triangles, or sorry, the diamonds. I want them to be triangles, but they're not. Moving the diamonds across the membrane from the outside to the inside. So we're from the low concentration to the high concentration. Now, in order to do this, we have to use ATP, so adenosine triphosphate. Now, if you guys remember from biology, ATP is the energy currency of the cell. It's the type of energy it prefers to do things. Okay, so ATP is going to attach to the protein. What's going to happen is we're going to cut off one of those phosphate groups and go from ATP to ADP, adenosine diphosphate. And the breaking of that bond is going to power the movement of this solute from the area of low concentration to high concentration. It's going to cause that shape change and going to power things to come across. Okay, so we've talked a lot about transport, passive and active. So what might a bacterial cell want to transport either into the cell or out of the cell? So take a minute in your notes, write down a list, and here in a second we'll go through the answers I came up with. Okay, so let's talk about what a cell might want to transport. Well, easiest one, nutrients, right? They want to bring in things that are going to help them survive into the cell. So whether it's sugars like glucose, nutrients like amino acids, they were going to want to bring them in the cell. Okay, another thing that they'll want to bring into the cell is amino acids. Okay, they need to make proteins. Proteins are the lifeblood of the cell. They need to have it in order to function correctly. So in order to make proteins, we're going to have a bunch of different amino acids. Okay, and some other things that I thought about are iron. Iron helps certain bacterial cells work properly. Other minerals, so we can think about ions. So sodium plus, chlorine minus ions, the H plus hydrogen proton ion. Okay, so these are things that they would want to bring in. Now, what would they like to take out? Well, easiest one, waste. Any waste byproducts they'll want to get rid of. Because if those waste products build up in the cell, it's going to change the inside environment. So the proteins are going to start to denature, things are going to break down, and the cell's not going to survive very long. Okay, and we just talked about this one, toxins and antibiotics. So for some, toxins are their defense mechanism. So like Vibrio cholerae, they're going to release toxins, and they're going to want to get rid of things that are toxic to them. So like antibiotics. Okay, so these are some things that the bacterial cell might want to bring in and out. Okay, so we've talked about how ATP can be used to power active transport. Let's talk about a specific example. So one of the ways that active transport can be beneficial to the bacterial cell is that certain species of bacteria have learned to, or evolved to pump antibiotics out. Okay, so these antibiotic pumps, these effusion pumps, as I believe what they're called, are going to need ATP in order to send the antibiotic against the concentration gradient. Because inside the cell, there's not going to be a high concentration, but outside the cell is going to be a large one. Okay, so the way some of these work, especially in the Lactococcus species where we see this a lot, okay, we're going to have a membrane protein called a substrate binding protein. This guy's important. He's going to help the substrate or solute so that thing that needs to get inside the cell come in, okay, or go out in the case of these antibiotic pumps. Okay, so the substrate binding protein is going to bind to the solute, okay, and it's going to make this binding protein solute complex, okay, and it's going to come and then attach to the transporter. Okay, once it binds to the transporter, it's going to cause a shape change. Okay, so it's going to open this channel up, but in order to bring this from an area of high concentration to an area of low concentration, what's going to happen is ATP is going to bind to this side, okay, and then it's going to cleave one of those phosphate groups off and make ADP, and the energy formed in that or stored in that bond is going to open this up and allow the solute to come through. Now for antibiotic pumps, we're actually going to go in the opposite direction. So in this illustration, they're coming in. Okay, so these transporter proteins are called ATPases. Okay, they're a special enzyme that's going to bring things in and use ATP to power that. Okay, so for the antibiotic pumps, instead of bringing things into the cytoplasm, it's going to be in the opposite direction. It's going to try to get them out. Okay, when a bacterial species can pump these antibiotics out, this is going to be what we call a method of pathogenesis. Okay, so it's going to allow the pathogen to keep making you sick, but it also makes it resistant to those antibiotics, so it can evade that treatment. Okay, so we're going to watch a animation. So let's watch that.
Transporting nutrients and wastes across the cell membrane often requires energy, using a form of transport called active transport. Most nutrients are present at lower concentrations outside the cell than inside, so the active transport further concentrates the nutrients inside the cell. Movement by active transport requires spending energy. In contrast, when a substance is transported from a higher concentration across the membrane to a lower concentration, the process does not require added energy. This is passive transport. In passive transport, a substance spontaneously moves down its concentration gradient. When a membrane protein provides a passageway, the passive transport process is more specifically called facilitated diffusion. Facilitated diffusion can move a molecule into or out of the cell. The net movement in one direction or the other depends on the direction of the concentration gradient. Consider that a concentration gradient represents a form of energy. As a substance moves across the membrane, down its concentration gradient, the energy of its movement can be harnessed to do additional work. For instance, it can fuel the active transport of another substance against its concentration gradient. The use of energy from one gradient to drive transport up another gradient is a type of active transport called coupled transport. When the two substances travel in the same direction across the membrane, the coupled transport is called symport. When two different substances are moved in opposite directions across the membrane, the active transport process is called antiport. In this example, the antiporter binds to substrate A on the cytoplasmic side of the membrane. As the antiporter opens to the outside, substrate A is released down its concentration gradient. Now substrate B can bind. The antiporter opens back to the inside of the cell, where substrate B is released, against its concentration gradient. Another way cells fuel active transport is by the use of ATP to move solutes uphill against their concentration gradients. The largest family of energy-driven transport systems are the ABC transporters, named for their ATP binding cassettes. There are two main types of ABC transporters, influx and efflux. Influx ABC transporters carry critical nutrients such as sugars, amino acids, and minerals. Efflux ABC transporters expel hazardous wastes and are generally used for removing antibiotics from the cell. An ABC transporter consists of two proteins that form a membrane channel and two cytoplasmic proteins that contain a conserved amino acid motif that binds ATP. An additional protein, called a substrate binding protein, initially binds the substrate, also called a solute. Substrate binding proteins have a high affinity for their matched solutes, so their use increases the efficiency of transport when concentrations of the solute are low. The substrate binding protein directs the solute to the channel protein. Hydrolysis of ATP to ADP plus phosphate yields energy to open the channel, allowing the solute to enter the cell. The transporter returns to its resting state.
Okay, so that animation was pretty important. So this last slide in this lecture, we're going to talk about it really quickly. This is something that is going to come back when we talk about metabolism, so it's kind of important to know. Okay, so we have a lot of different concentration gradients in cells and in our bodies, but for bacterial cells, one of the most important concentration gradients deal with the hydrogen 1 plus ion, or the proton. Okay, so the hydrogen 1 plus gradient is known as PMF, or the proton motive force. Okay, so these H plus ions are going to power quite a lot of things. Okay, mostly H plus is used to make ATP, so it's something that powers what is called ATP synthase. Okay, we talked about this in regular biology when we talked about cellular respiration. So our hydrogen gradient, we talked about our proton motive forces in our mitochondrial cells. So we have the inner and outer membranes of our mitochondria. The mitochondria is the powerhouse of the cell, so that's where we make energy. So we have ATP synthase that's in these mitochondrial membranes, and we use H plus to power the ATP synthase. And we use the electron transport chain to generate the H plus concentration gradient. Now, with eukaryotic cells and things that have mitochondria, like we do, cellular respiration is very effective. We're going to make between 36 and 38 ATP by the time we're done with cellular respiration, and most of it's going to be done in that last step using ATP synthase. Now, bacterial cells don't have mitochondria. They have no membrane-bound organelles like the eukaryotic cells do. Okay, so their ATP production is going to be directly in their plasma membrane. Okay, so instead of shipping it out into an intermembrane space like our mitochondria do, these H plus ions are going to be pumped outside of the cell. Okay, and then so they're going to go and create this gradient. So they're going to create a high concentration outside of the cell, and a low concentration inside the cell. So what's going to happen is these H plus are going to then go through this channel in ATP synthase. It's going to cause this part of ATP synthase to turn. So in bacterial cells, it's kind of like a ratchet. So this is going to turn, and it's going to take an inorganic phosphate and ADP and combine them to make ATP again. Okay, and then that's going to create energy for our cell. Now, where our mitochondrial cells are really very efficient at this, bacterial cells aren't, because one of the things that's happening is since we're pumping this outside of the cell and we've created a concentration gradient, these H plus cells aren't just going to come back in through ATP synthase. They can come back in through any old any old channel protein. They can get lost. So it's not as efficient as using our mitochondria. It still is enough to produce energy, but not as much. Now, this whole process sometimes is targeted by different medications and different antibiotics because bacterial ATP synthase is different than our ATP synthase in our cells. Okay, so whenever we have something that's different in bacterial cells or in a pathogen versus ours, that makes it a prime candidate for antibiotic therapy. Okay, so ATP synthase is one of those. Okay, that's section 5.2. We've talked about the different functions of the cell membrane in bacteria. We've talked about active transport. We've talked about concentration gradients and passive transport. Those transporters we talked about are specifically called ABC transporters, as you saw in the animation. They're going to move things from a low concentration to a high concentration. So this is usually referred to as against the gradient or uphill against the gradient. And then we talked about the proton motive force. Okay, so this proton motive force not only powers ATP synthesis, but it also is going to help those transporters, those efflux ABC transporters, to pump the toxic antibiotics out of the cell. So that's another function of the proton gradient. Okay, so that is chapter 5.2. We'll see you for chapter 5.3.